of DNA and RNA, these nucleotides that are out there, remember these are polymers that we're used to thinking about, and they are made of these little building blocks. And when you look at the building block, the nucleotide monomer, it does contain a 5-carbon sugar right here that has a phosphate group attached to it, and it also has this nitrogen-containing base attached to that sugar right there. And when we look at this sugar, this is where we see one of the differences between these two different types of nucleic acid. For your DNA, the reason it's deoxyribose is that pentose sugar is missing an oxygen hanging off of this second carbon right here. And if you look at RNA over here, you can see that the second carbon in that ring structure actually does have a hydroxyl group right there. And for those of you who think this doesn't make much of a difference, it's just a single oxygen there, well, it turns out that DNA is a lot more stable molecule compared to RNA because of that extra oxygen on your ribose sugar there. And RNA polymers will actually break down if you mix them up in hot water with an alkaline pH. It'll take that polymer and break it down to these nucleotide monomer structures here. And the other thing when we look at that is, remember this nitrogen-containing base that we have over here. And if we look at the different nitrogen-containing bases that are out there, they fall into two classes. Your purine residues that actually have this double ring structure, and that includes adenine and guanine. And then your pyrimidine bases that are out there, including cytosine, thymine, and uracil. These have a single ring structure. And again, you can see that there is this nitrogen in there. And if you remember back to our chemistry stuff, when you look at molecules that contain oxygen and nitrogen, those are going to be polar elements. And that means that the electrons are going to spend some more time around these elements, and that's going to result in a little bit of polarity to their covalent bonds. And when you have polarity to covalent bonds, that makes them like little magnets, and they're going to be able to start forming hydrogen bonds with other elements that are out there that also have that polarity or electronegativity built to those atoms. If we take a look at taking these nucleotide monomers and turning them into polynucleotides, this is where another side group on that sugar chain makes a big difference. And if we look at this right here, here we have our sugar with our phosphate hanging off of it and a T residue. And you can see this is coming off of the 5' carbon. You have your phosphate group right there. And if you look at this, the next base is actually attached to that. So here we have a 3' hydroxyl group hanging off the side of this pentose sugar right here. Not the 2', now we're looking at the 3' carbon. And there you have a phosphate group attached, and there is your next sugar, and then your phosphate, and then your next base, and we have a free 3' hydroxyl group hanging off there. And this is the site where the next nucleotide would be added to this growing chain or growing polynucleotide right there. And what people refer to is this gives us a sugar phosphate backbone right here, and your nitrogen containing bases are basically hanging off of that sugar phosphate backbone. And if you take a look at the structure of your DNA there, the ribbons that we see in this diagram, there is your sugar phosphate backbone, and then your bases are projecting out here. And if we take this double helix right there and untwist it, we can see the flat version right there. There is one strand of your polynucleotide for DNA, and then we have the other strand. And what people found is that these two strands actually go in opposite directions. There's our 5' phosphate end, and here's our 3' hydroxyl group right there. And this is where your new bases would be added. But if you look at the opposite strand over here, notice the 3' hydroxyl is on the opposite side there. And so this is what makes DNA strands known as anti-parallel. They go in opposite directions like a two-way road out there. If you look at RNA, it's typically a single-stranded molecule, not double-stranded like this, and it's coated with proteins to help stabilize that interaction. But when you have DNA as this double-stranded molecule, it's very, very stable. If we look at these two strands right here, we also find that the two strands complement each other. And this was discovered by a Russian biochemist who was able to actually take DNA and purify it from different cells that are out there, and he was able to chemically hydrolyze the DNA. He mixed it up with some acids and then put it under high heat conditions. And what happens then is the DNA actually breaks down into the monomers, and then he took the resulting mixture there and he measured the amount of the 4 different bases that came out. And what he found is the ratios of these bases were always consistent, even when you looked in different organisms. The amount of A was always equal to the amount of T residues, and the amount of your cytosines was always equal to the amount of the guanine residues. And this is what's known as Chargraff's rule. And what Watson thought about when he heard about Chargraff's data is that this is perfect because you can look at this as 2 strands for your DNA molecule, and they complement each other. In this strand, you can see there's a large base right there opposite the small base, and over here you can see small opposite the large, and that's going to result in these 2 strands having the same width when we're looking at this molecule. And that's really important, as we'll see later on, because this allows proteins to be able to bind this and basically feel their way along the DNA right there. The other thing that is really kind of interesting is that we can actually take DNA and denature it and separate the 2 strands that are out there. So in this little figure, we can see there is your DNA in its normal native state with the double-stranded DNA molecules that are held together by the hydrogen bonds between these complementary bases. And if you take them and heat them up and mix them up with an alkaline solution, which you remember that would break down RNA, but really what happens with DNA is it separates the 2 strands, and now you end up with single-stranded DNA. And one of the amazing things about it is these 2 strands, as they cool back down, are actually going to re-anneal, and they will reform those hydrogen bonds between their complementary bases. And what people found is that if your DNA is really rich in C's and G's, then it actually is harder to denature. And if you think about that, C's and G's form 3 hydrogen bonds, where A's and T's only form 2. So if you have a higher percentage of C and G's in your DNA, it's going to be harder for your DNA to separate. And if you look at the hyperthermophiles that are out there, they tend to have a little bit more C's and G's in their chromosomes, because that is one of the things that helps to hold the 2 strands together. The last thing we want to take a look at is some of these terms that are associated with genetics that are out there. And if we look at a gene, effectively what a gene is it's a segment of nucleic acids that contains a code to make a functional product. And what we're used to thinking about, and we learn in a lot of biology classes, is that DNA codes for messages that are going to be used to make your proteins. But if you think about a virus that does not have DNA, it's just RNA, for them, a gene codes for a functional product. In this case, it's coding for a protein, so they don't ever have DNA in there. So if we use this definition that a gene is a segment of nucleic acids that codes for a functional product, now we can cover our cells with their DNA, but also our viruses that lack DNA. If we work our way up, well, where do you find these genes? They're going to be on your chromosomes. And this is just a structure that is composed of your packaged DNA with your genes on it that are coding for those functional products. And if we work our way all the way up and start to look at the genome, now you're looking at the sum of all the DNA and all the genes inside of this cell that give that cell its own distinctive genetic makeup there. And if you think about that, those chromosomes that are containing all of these genes and potentially other DNA outside of those chromosomes, that is what gives us our genotype, which is that genetic makeup for the cells, and that genotype is what we see in what's known as the phenotype. So you express the genes in those cells, and that's going to result in specific structures and functions that ultimately are going to come from the proteins that are being encoded by the genes on your DNA, and that's going to give you that physical makeup for your cells. And when we look at the genomes for cells out there and look at all that genetic material that we find with cells, we do see some very distinct patterns out there. Your viruses that are out there, those can have really, really small genomes with a few thousand bases, all the way up to these viruses that mimic cells that are just on the edge, and they almost have as much DNA as what we would find in our standard kind of E. coli in the lab. When you look at these things, you're looking at things that can have as few as 2 up to more than 2,000 genes for a virus that is out there. When we look at our bacteria with these prokaryotic cells, remember they have 1 or 2 circular chromosomes that can have millions of bases in them. And if you remember, those chromosomes are also attached to the cell membrane and are in what's referred to as the nucleoid region of the cell. And these bacteria genomes can range in size from 500,000 to 14 million bases. And if you look at your E. coli, it averages around 5 million bases, and we're looking at about 5,000 genes depending on the strain that you're looking at there. But the interesting thing is if you took that chromosome and were able to unwind it, it would stretch out to be about 1 millimeter long. And E. coli is 1 to 2 micrometers. So that DNA that makes up the chromosome is actually about 1,000 times longer than the cell itself, but it is supercoiled to fit into that nucleoid region. And remember, there can also be extrachromosomal DNA, viruses or plasmids that can be found floating outside of the chromosome in the cytoplasm there. If we switch over to our eukaryotic cells and take a look at their genomes, remember these ones have linear chromosomes, and they will have more than one. If you look at a yeast cell that's out there, you're looking at something that has 4 linear chromosomes and 12 million base pairs there, more than 5,000 genes. So longer genome, but also more genes compared to your standard bacteria that we use in the lab. Also remember, when you look at eukaryotic cells, they do have organelles like mitochondria and chloroplasts that can also contain DNA that can affect the cell. So this is part of the genetic complement of the cell there. And when we look at our standard mitochondria, it's a little over 16,000 bases long, and it's got 37 genes on it. When we look at some eukaryotic cells that are out there, they can also have plasmids, and we found those in fungi and protozoan cells. They actually do have this extra chromosomal DNA floating around in the cytoplasm. When we get up to ourselves here and we look at humans, we're looking at 3 billion base pairs with more than 20,000 genes that are on 23 pairs of linear chromosomes. And when we look at the chromosome structure in eukaryotic cells, what we find is the DNA is wrapped around histone proteins to form what are known as nucleosomes. They're referred to as beads on a string right here. And then those beads on a string are wrapped up to form chromatin fibers. And when you look at the chromatin fibers, some of them are going to be tightly packed together, and that tends to have the DNA that's not being used. But if the chromatin fibers are loosely packed, then that DNA is actively being used. And we really only see this chromosome in this highly condensed state when the cell is dividing and preparing to divide and going through the process of mitosis or meiosis.